Hi, everyone. Welcome to the second part of the session of Deep Learning in Science. So we're very excited to have our first speaker of this session, Eero Simoncelli, the Silver Professor of Computational Neuroscience at NYU, a Fellow of the IEEE, and Howard Hughes Medical Investigator.
Thank you, Maithra. And thanks to the organizers for inviting me. I'm delighted to be here. This has been fun so far. So I thought it's about time in the meeting for somebody to get up and describe something that deep nets cannot do, at least at this moment in time. So I think that's my role. I could have talked about engineering or neurobiology or perception, but I'm going to stick with perception for today. When that image that was on the previous slide comes into your eyes, it goes into your brain and into your eyes first, and then it makes its way back into your brain, gets processed by first your retina, then something called the lateral geniculate nucleus, a little way station here, and then goes back to your visual cortex, where it gets sort of torn apart and transformed into a format that allows us to understand what it is that we're looking at. And that miracle of vision, that somehow vision arises from that cascade of processes, is what I study in my lab. And in general, we're interested in really three kinds of questions. And the three questions, I guess if I have to summarize it, they go something like this. How do populations of neurons encode visual information? How do those representations enable and limit the perception that we have? And how can we use those ideas and those principles to improve engineered systems? Okay, so as I said, I'm going to focus mostly on the second question, but there will be little sprinklings of the other two along the way. So my existence was thrown into disarray in the last kind of 5 years by the emergence of deep neural networks. On the one hand, everything looks very familiar to me. This is really what we've been doing all along. Ever since I was a PhD student, I've been doing things that are about layered representations and stacks of things that have convolution in them and nonlinearities. But I've been thinking about those as sort of simple models, maybe just a few stages, to describe neurobiology, to describe perception, and also to build solutions to engineering problems, as I already mentioned. Deep neural networks, I think what I'm going to say about them right at the moment, I think is fairly uncontroversial. First, they're largely inspired by neurobiology. That's where a lot of the ideas originally came from, and we've already heard that this morning from a number of speakers. Second, that the engineering results that they've produced are really astonishing, although quite brittle, and I think I and many of us in this audience know that, and we know that there are examples of failures, sometimes catastrophic failures, and those are, from a scientific perspective, when you encounter a catastrophic failure, that's interesting. In science, you don't prove things, you disprove them. You look for failures. You know that all the models that you ever come up with will be wrong. And so what you're doing is trying to find out, are they useful? Do they account for some of the data? Do they account for the data under certain conditions? You're not trying to prove that they're correct. And so when you encounter a failure, you look at that as an opportunity, as something that you can learn from, something that gives you a chance to think about how to improve the system, and you don't hide it or run away from it or sweep it under the rug. And my sense is that a lot of the engineering community, when they encounter these failures, they quickly look for a way to cover it up. Sorry if you're in the engineering community and you don't view it that way, but So I just, I'm not going to show, well, the failures I'll show have to do with modeling perception. So if you're in the engineering community, you can say, well, I don't care about perception, so I don't really care if it fails to explain perception. Okay, so let's continue. So as a model for sensory neurobiology, I think that deep neural networks, of course, provide, because they're inspired by neurobiology, they do provide us some framework maybe for thinking about neurobiology. And there are some really nice results from, for example, Yamins and DiCarlo, some more from Nico Kriegeskorda, showing similarities and relationships between stages of neural networks and populations of neurons, or MRI responses to populations of neurons in Nico's case. And I think those things are a really nice starting point for understanding how we can relate these things. But there are also a lot of things missing in these models. They're not going to really provide a proper scientific description of the neurobiology of the visual system or the auditory system or really any brain system. They're largely trained with supervised, the biology is, as Tommy pointed out and as many people have pointed out, largely trained with unsupervised learning and with objectives that are surely not about identifying some discrete class of objects. Biology uses local learning rules that are not just done in some sort of initial phase, but are constantly at work. Our brain is constantly adapting, adjusting, recalibrating, gain controlling, and adjusting responses throughout the entire brain. It involves a lot of recurrence. Of course, that's starting to appear in deep neural networks, but the recurrence in the brain is something that is, I think, much more sophisticated and more widespread and ubiquitous. And last of all, of course, there are myriad biophysical details which are not shared by deep neural networks, and whether those matter or not computationally is something that I think we don't know the answer to. Okay, but in any case, there are a lot of differences, and some of those differences are surely going to matter in terms of scientific progress. And then the last point I'm going to make, whoops, so we could say that both for the engineering and for the sensory neurobiology, the glass is sort of half full and half empty at the same time, and we should try to keep a balanced perspective and push back a little bit against some of the hype and the press, let's say. Okay, and the last point is whether we can use deep networks to sort of describe or model perception, and that's kind of what I'm going to focus on for the talk. I'll mention a little bit about gain control, and I'll say some things about perception. Okay. So, deep networks are built on this idea that you can stack some sort of fundamental canonical unit of computation, and that by repeating that in layers and also spatially or in terms of time, that you can actually get a lot of power in the computation. And I think that in some sense it's sort of obvious that we already know, because of sort of universal approximation theorems, that if you combine enough such elements and if they have a little bit of nonlinearity in them, that's sufficient to guarantee that you can match any function. And I think so the question sort of becomes not whether we can describe sensory systems this way in terms of these kinds of canonical elements, but really how simple can we make those elements? So I think the deep neural network literature has sort of collapsed, in a sense, into a mode where there's a belief that you can use the simplest possible element, a linear thing, followed by some sort of rectifier. And that's, if you like, that's the simplest form of nonlinear computational element, and that's the one that almost the entire field has now collapsed on, because it's been basically studied and found that it works well in practice. I think, in fact, neurobiology uses something quite a bit more sophisticated than that. Just kind of, this little quote reminds me that, you know, you could say that this is really a good thing, because simplicity is a good thing. We should go for the simplest possible model, and it is remarkable how well that very, very simple element performs when put together in these kinds of architectures. But I think, you know, the biology suggests that there are, or gives us clues, that maybe we ought to be including something a little bit more sophisticated, and maybe that would offer us a lot of computational advantage. So I'll try to show you two simple examples where I think that that's the case. Okay, so the first example is a problem of trying to quantify perceptual distances. So let me explain the problem. If I have an image, let's say this is an original image and this is a distorted image, which I hope you can all see. It is actually a distorted version of that. We'd like to know we'd like to have some measure, some number basically, that quantifies how bad that distortion is. And we know that the standard way of doing this in all of engineering for the past, you know, sort of 60 or 70 years is to measure mean squared error. That's the Euclidean distance between, you know, all of these pixels and all of these pixels. Literally take the difference, the pixel-wise difference between this image and this image, square those differences, add them up, take the average, right? So the mean squared error is a perfectly, it's a beautiful thing. We know that an enormous amount of our engineering and mathematics rests on things like Euclidean distance, but it actually doesn't work very well for this problem. So here are 8 images surrounding that middle one, which is the original, and they all have the same mean squared error, and I hope you can all see, even if you're in the back of the room or if wherever you're sitting, that these are not all equally bad. Some of them are really bad, and some of them look pretty good. Some of them might even look better than the original, like this one. So you can see something like that. It looks pretty terrible, and like I said, this one looks really good. So mean squared error is not a good way to measure this. At least it is not matching what we see perceptually. So why does it fail? Well, the human visual system does all of this decomposition, transforms things into some internal space. And it's the nonlinear processing that goes on in that transformation, coupled with the noise, that really determines your ability to discriminate and detect differences between images. So what we need, if we want to describe this in a more perceptual way, is to try to somehow mimic that process, or mimic the most essential elements of that process. And so what we might imagine as a first pass would be that let's use mean squared error, Euclidean distance, but let's do it in some other space. Let's find some function f that we can map our images through into some internal space. We can think of that as maybe a population of neurons that are representing the image, and then we'll measure the errors in that space. That's not the same as measuring back in the image space, because this transformation is nonlinear. If it was linear, then it would be basically equivalent. So how do we choose f? How do we pick a function? So we set out to do this using a very, very simple model. This is a model that's hand-constructed, unlike the sort of deep net approach. We're not learning things here. It's based on similar kinds of operations. So that's a linear filter. It's a center surround filter that often gets used to describe processing in the retina or the early visual system. This is another center surround filter. There's an on filter and an off filter. These things are low-pass filters, these disks. It's just a cartoon, of course. These are disks that are computing low-pass or averages of local image intensities. And then we divide this thing by this thing. We call that luminance gain control. And we know that this is a property of the early visual system, or pretty early on in the retina. And we think that it's actually quite important for the system to be able to process this sort of high dynamic range of natural images that occur in the real world. Okay, so that's followed by another stage that does another form of divisive local gain control, and this one is contrast gain control. So we have a luminance gain control and a contrast gain control, and these kinds of elements are not the kinds of things that you see in deep neural networks. This is different from a deep neural network in that, although it shares the convolutional sort of structure, it's using nonlinear operators that are pretty different than what you find in most deep nets. It's also very shallow. This is basically, if you like, 2 stages of processing, depending on how you count. Okay. So that's a simple model for early visual system. And what we're going to do is we're going to try to fit data. The data are coming from a database, this TID 2008 database, which is a large database of human responses on quality of images. Their ratings, there are something like 500,000 ratings in there from, I have it written down here, 800 observers over 1,700 distorted images. So it's a lot of data. It's a pretty large study. And what we're going to do is we're going to fit this model. This LGN model has, I'm going to call it an LGN or an on-off model. It has 12 parameters because we're going to control the sizes of all those filters and a few little parameters that have to do with the nonlinearities. So there are, in the end, 12 parameters. We're going to compare that to two things from the deep net world. One is VGG, which is a standard deep net trained on object recognition. We're going to take a weighted sum of the Euclidean distances, the mean squared error, at 13 different stages of VGG, and we're going to combine those. So the weights of that sum are the things that we're going to learn. So there's 13 parameters. Okay? So these two have similar parameterization, 12 and 13, and then we're also going to compare to a generic convolutional neural network. I can't remember how many stages. It's something like 6 or 7, and there are 500,000 parameters. Okay, so we're going to fit all of those to the human subject data in this database, and we're going to ask which one can explain the data best. And I don't know whether the answer will surprise you or not surprise you, but it basically is that it's a tie. So here's, these are correlations with the human data. It's cross-validated, so we're not, to avoid any kind of overfitting. And what you see is that the performance of mean squared error is pretty poor, as I already showed you with that slide earlier. And the on-off model, the VGG model, and the CNN model all perform quite well. These are actually getting close to the sort of level of noise in the data. This is about as well as you're going to expect to do on this data set. So we're getting close to ceiling, pretty close. I think you could probably get a little bit higher if you look at the noise, the ability of humans to predict each other. But in any case, this is close to ceiling, and the performance is very, very similar. If anything, the CNN wins, but by only a very small margin. And we could just stop there and say, well, we'll declare victory for the on-off, the sort of biological model, because it was the simplest one. Or we can say, well, no, it's a tie. Or we can say it's a victory for the deep nets because they were complete. This CNN on the right was completely learned from the data. We didn't impose anything other than standard convolutional neural network structure. So you could decide for yourself how you'd like to interpret this result. We didn't want to do that. We wanted to go beyond this, so just cross-validating on that same data set only can tell us so much, because all that data came from the same kinds of distortions and the same kind of database of images. So what we wanted to do is to take each of these models, think of it as a scientific hypothesis, and ask what, or theory, and ask what prediction does that theory make. So if you take each of these models, you can ask, what can I predict about what I might see in some other circumstance, and can I test those predictions? So what are the predictions? Here's the idea. Here's an image represented in a two-dimensional space on this screen. So I'm only showing you, for example, 2 pixels, and the value of 2 of the pixels. The actual image is, let's say, 1,000,000 pixels, so this point really lies in a 1,000,000-dimensional space, but I'm just showing you 2 of those dimensions. The idea is that each of these models takes that pixel space and transforms it into some internal model space. So there's another vector here, call it R for response, that lies in some response space, which is also very high dimensional, and we don't have to worry about the exact dimensionality for the description I'm about to give you. The idea is that we're going to treat errors or distances in this space as perceptually uniform. That is, all of those points on that orange circle are equally bad in this model. And what we want to do is pull that back into the original space, and there are mathematical tools that we can use to do that. Some of you know about those tools. If we were doing this properly using a noise model in this space, then we would be using the Fisher information matrix, or we could just be looking at the Jacobian of the transformation. So there's a little bit of technicality for those of you that's meaningful. But if that didn't mean anything to you, don't worry about it. The point is very simple. We have a nonlinear transformation. This is the set of all images that are equally bad in this space. That's how we're going to define our model. And we can pull it back, and that induces a set of equally bad images in this space, which is not uniform. And in particular, what we're going to be interested in, because remember, this is really a 1,000,000-dimensional space. I'm only showing you 2 pixels. It's a 1,000,000-dimensional space. What we're going to do, because we want to test this on human subjects, is we're going to find the best and worst case, the extrema in this space. We're going to find the direction that has the largest distance in this space, and the direction that has the smallest distance. And those two are going to be, if you like, stimuli or perturbations to the original stimulus X, which are going to be most or least noticeable according to this is a prediction of a given model. We can make these predictions for each of the models, and then we can compare them by actually asking human subjects how visible are those distortions. So let me walk you through what that looks like. We can first get a baseline by asking human subjects. We have I think we have 19, yeah, n equals 19 human subjects. This is the average visibility of just white noise. It's not the visibility, I should explain the axis. On this vertical axis is the threshold, that is, how strong do we have to make the noise, how big do we have to make its amplitude in order for a human to reliably see it. Okay, so this star represents that value. If we now ask each of our models to generate, we go through a computational procedure to generate in this case, the least noticeable error according to that model. We get, for this parrot image, we get 3 different answers. The on-off model, the biological model, produces this sort of funny, lumpy-looking bit of noise that is kind of aligned with the contours and the structure of the image. And the 2 deep neural networks are producing something that looks kind of like white noise. It's not very structured. So we can measure the visibility of those 3 kinds of distortion by asking our human subjects, basically, how strong do we have to make it for them to see it? And the answer is shown here in these data. This is, again, averaged over 19 subjects. The on-off model, this distortion, is very hard to see. We have to make it very large. We have to push it up to an amplitude of about 20 for the humans to reliably see it. These other ones are pretty easy to see. They're almost as easy to see as white noise. They're slightly less visible than white noise. Okay? And then, of course, the other extreme, we look for the most visible distortion, and each of these models, again, produces a prediction. We are optimizing that prediction. We're finding the image perturbation that is most visible according to this model, most visible according to this model, most visible according to this model. When we add those to the image and we ask how much do we have to add for humans to see them, we get these, and this is our answer. We find that the on-off, this model, this distortion generated by the on-off model, the biological model, we need only a very, very small amount for humans to reliably see it. It's down here at something like 0.05 is the amplitude. magnitude. And for these, they also need less, for example, than the white noise, but they're quite a bit above what we get for the on-off model. The important thing is that the range here from the least to, from the most to the least visible for the on-off model is really enormous. It's a factor of about 1,000, about 3 orders of magnitude, whereas these things are about a factor of about 15. This is a log scale. So we have really a much, much stronger predictive power coming out of this simple biological model than we do from either of the deep nets, at least in terms of this perceptual task. It's a very simple perceptual task, very basic. We don't know what part of the brain our observers are using when they solve this task, but we do see from this that even an early visual model that's just basically describing your retina, sort of early processing before it even gets to your cortex, is enough to capture a fair amount or to make predictions that are quite strong in terms of what humans can see or not see. Okay, so, So that one last point is just that if we modify that biological model and try to peel apart the pieces of that biological model and ask what is it that matters here, we can take the full model, we can back it off to a single, instead of 2 different filters, we can just go for 1 filter, or we can back off and get rid of the contrast gain control and just use the luminance gain control. These 3 models are those 3 models, and you can see they get consecutively worse. The points are getting closer and closer to each other, but it's only when we go all the way back to a simple linear nonlinear model, that is a linear filter followed by a simple rectifying nonlinearity, that we see that the result really collapses. So from this, I guess I would say that these models all have some sort of local gain control, and the local gain control is actually critically important for these results. And that's the piece of this that's kind of not part of the canonical operation that you see in most deep neural networks. Okay, let me show you another quick example in the remaining, I have about 10 minutes. Okay, so we've been working with a theory for a little while now that the human brain is really optimized to make predictions, and the visual system in particular is optimized to make predictions about what will come next visually. One way to think about that problem is, it's kind of hard to think in general about predictions, but one concept that emerged when we started to think about this and how we might test it is the idea that the representations that you have internally are somehow taking the input, which follows very complicated trajectories. The trajectory of visual images, if you think about what the pixels are doing, are actually quite complicated. They follow very curved and complex trajectories. So there's a simple concept, which is that what you do with your brain is you transform that input into some space where the trajectories are straighter as in straight lines, and therefore more predictable. So it's a simple and almost naive concept, and it's something that I have to admit, when we first started thinking about it, I thought it was unlikely to work. But the nice thing is that it gives us something that we can test. So the idea, again, is in the space of all those pixels, a million-dimensional space, if I follow a temporal trajectory of, let's say, a video, it follows some very curved path. But if I look inside the brain, if I could look at a population of neurons that was representing this video, or if I could measure this somehow perceptually, what I would find out is that the path is much straighter and more predictable. So we wanted to test that, and I should say that we're actually now testing this in terms of actual neural measurements from macaque monkeys, but for the time being, I'm just going to show you the perceptual results. We basically worked out a way to measure this perceptually. I don't have time to detail how we do that, but we can basically ask human subjects in the course of about an hour and a half of making them compare different images, frames from a short movie. We can actually get measures of distance between the frames, and we can use those measures of distance to actually determine the curvature of representation within their brains. We can measure the curvature in the image space very easily. That's just a matter of using a discrete measure of curvature, which has to do with the angle that's subtended as you follow what these points are doing as they go through the pixel space. So you basically can measure with using dot products and standard linear algebra tools. You can measure the angles that are subtended at each of these little kinks, and you can take the average over the video, and that gives you the average curvature of the sequence. So we can measure the average curvature in the pixel domain, the input. We can measure the average curvature in the brain using these perceptual experiments that I, again, don't have time to detail. But the idea is just by asking people to make comparisons between pairs of images, we can measure distances internally, and from distances we can get angles, and from angles we get curvature. Okay, so that's sort of the quick nutshell geometry. All right. So now we can measure curvature, and what do we find? So here's an example. This is 3 frames of a movie sequence. That's the first frame. That's the last sequence. This is a shot in a forest. The camera is moving fairly close to a pile of leaves and sticks. And what we find is that if we look in the pixel domain, this is projected into 2 dimensions again, so you can visualize it. This is a very curved path, this sequence. But if we actually take the perceptual measurements that we get out of our human observers, it's actually almost straight. So for this sequence, it works quite well. And what you see on the right is a histogram over and sort of measured over, let's see, 12 different sequences and about 20 human subjects. So this is compiled data from all of those subjects and all those sequences. And what we find is, in general, this is a plot of the change in curvature that compared to a control condition, all of our subjects across essentially all of these sequences show a reduction of curvature in their brains compared to the curvature of the original sequence. So basically the hypothesis seems to hold up pretty well. Humans are reducing the curvature of the trajectories of natural videos in their brains. We, as a control, we wanted to compare what would happen if we took something that was straight in the pixel domain. So for that, we take this first frame and this last frame of that sequence, and we fade between them linearly. So by definition, this will be a straight line in the pixel domain, as seen here. This middle, you can see that this middle frame is basically the average of those 2, because that's literally what we're doing as we go through this sequence. The sequence is 11 frames long. I'm only showing you the first, the last, and the middle. The middle one is the average of the other 2. So what happens now when we test this on our human subjects is we find out that this sequence, which is very straight in the pixel domain, is actually much more curved inside their brains, according to these perceptual measurements that we're making. And we find that that holds up again across all of our subjects and across all of our images. In general, these sequences, these artificial sequences, are straight in the pixel domain and are increased in their curvature by the human brain. So it's the opposite behavior from the previous experiment. One more test we did, we looked at what happens if you take an image and you fade it in contrast. This is also straight in the pixel domain, and we thought, well, that's a pretty naturally occurring event. That should be something maybe that the brain continues to keep straight. That is, the predictions that it would make is that the contrast will continue to reduce, and sure enough, that seems to be roughly correct, that our human subjects do not change the curvature of these sequences. They're straight in the pixel domain, and they remain moderately straight or reasonably straight in the brain. Okay, so summarizing the results, it looks something like this. If we plot curvature change here in the pixel relative to the pixel domain, all of the sequences start out here at 0, and human perception spreads them out in the following way. The naturally occurring sequences that came from videos from video cameras are all being reduced in their curvature. The artificial ones are being increased in their curvature, and these contrast fades are being basically maintained at 0. And we can take simple models of the kind that I already told you about, that model for the LGN. We can tack on to that a model of visual cortex. And these are very, very simple models that capture what we think of as the essential elements of computation in those stages of processing. And we can ask, what do those models do in terms of curvature? And nicely enough, they actually take us in the right direction. The LGN model starts to straighten the natural videos. Sorry, that's the blue point. Straighten the natural videos, that is, reduce their curvature. It increases the curvature of the artificial ones, and it preserves the curvature of the contrast fades, and even more so for the V1 model. So nicely enough, this seems like the stages of processing that we see in the human visual system seem to go in the right direction toward explaining perception. They don't quite get there, but we have no expectation that your perception is entirely based on either this representation or this representation. We don't know what you're using in your brain when you solve our perceptual task. That is, when the subjects are sitting and doing the experiment, we don't know what they're using. Okay. So, and then the punchline for this meeting is that when you take a deep network that is trained for object recognition in this case, you can look at each of the individual layers and you can ask yourself, well, do any of these perform some sort of straightening operation as the human brain seems to do? And the answer is no. All of these, so just remind you, blue points are supposed to get less curved, the green points get more curved, and the red ones stay the same. In the deep net, sorry, There we go. In the deep net, they come out in the right order, but everybody is getting more curved. Curvature is increasing across all of these stages of the deep net and across all of the sequences. So at least for this particular problem, the deep net really doesn't behave anything like the humans. And I think that this is a pretty fundamental sort of behavior that we are uncovering about humans, about human perception. And so that remains a challenge to figure out, why is it that deep nets are not doing the things that humans are doing in this particular case. Okay, so just to conclude, simple canonical neural models, the kind that I've briefly shown you, and including things like local gain control, which is a kind of regulatory mechanism to keep everything operating in the right range, but I think maybe it has a more important set of computational roles that I don't have time to elucidate here. It allows us to predict human perception of visual distortions. It allows us to account for perceptual straightening of natural videos. Deep nets don't seem to be able to do either of those things, at least at the moment in their current forms. I think it's only a matter of time till we figure out how we can maybe get them or get something like them to perform these things. But at the moment, they don't. And I didn't talk about either of these things. These are both engineering applications. You can use these kinds of models to optimize image rendering for human observers. That's an engineering task. And you can also use them for compression. We've actually been able to get state-of-the-art compression results by using very shallow networks, a couple of stages with local gain control, and sort of with a lot more constraints that come, again, from biology. Okay, so this is just a thank you to the folks that did a lot of the work that I told you about. Their names were on some of those slides, but I didn't mention them before. That's Alex Berardino, who now works for Apple Computer. That's Johannes Balla, who now works for Google. That's Valero Lapara, who's back in Spain in a research science position in a university. And that's Olivier Henoff, who's gone off to Google also. And that's Robert Goris, who's taken a faculty job in UT Austin. And I guess, well, I don't have time because I think I've just run out of time. But this was my wish list for the deep neural network world. But maybe you can ask me a question about it if you want me to explain it. Thanks. Sorry, I went slightly over.
All right. Thanks, Eero, for a very interesting talk. And I think we have time for one quick question.
Is there a relationship between local gain control and batch normalization?
Yeah, thanks for that. A relationship. So batch normalization is used, for some of you probably know this, but in case you don't, batch normalization generally is used during training of deep neural networks. It is typically global, so the operation, what you divide by, what you normalize by is something like you subtract off the global mean of each of the layers and you divide by the local standard deviation. And then it is locked in place once you go to test the network or use the network. What I'm describing is quite different from that. It is local, so it's only, these operations are performed only on spatially local regions. It is about dividing by, if you like, you could think of it as the local standard deviation or the local mean, but it is local, and it is part of the network both during training and during operation.
But do they somewhat accomplish the same job?
In my opinion, it does not accomplish the same job, and I can give you detailed examples of that, but not in the time we have right now during the question. Thanks.